And today, sir will teach us about about pharmacognosy and the basics of pharmacognosy. So, without further delay, I will request to sir to start the session. Please, sir.
Thank you, sir, for this introduction. With your permission, I would like to share the presentation now. So,
Sure, sir. Sure, sure. And I would expect to from the students that everyone will be interactive to sir, and don't hesitate to ask any question.
uh, is it visible?
Yes, sir. Yes, sir. Is it clearly? It is clearly visible. Sir, can you please move it to the slideshow mode?
Yes, just trying this.
Okay. Okay. Okay, sir.
Yeah, is it visible now? Is it completely in slideshow mode?
No, sir. Please click on the slideshow and start from the current slide, so it will be visible. Sir, I think it is fine. So you can kindly continue, sir.
I'm just trying. Just
Okay, sir. Okay. Yeah, yeah, yeah. From current slide. Yeah.
now, is it visible? I don't think it's visible.
No, sir. It is not visible till now.
Yeah.
Okay, so no problem. If you can change the slide through the lecture, then it is not a problem for us.
Okay. Just let me know if the slides are changing or not. I will just start now.
Definitely, definitely, definitely. Okay, sir. Okay, sir. Take it.
Okay. Uh, Good evening, everyone. I think everyone here is a student, so I would try and share my experience with my experience of last 9 years. What I have been teaching in different colleges. So uh, I have done my masters in pharmacognosy. So I know. You have been studying botany, and there is a very important part that you will be studying, that is pharmacognosy. Now, without pharmacognosy, our branch, our pharmacy department, it is almost impossible to run, because whenever we think about medicine, we think about drugs, we always think about the historical aspects and how it has come to being. So whatever we have learned till now, from the earliest of the history, earliest days of history, history of humankind, we have seen people always go to nature to look for any kind of remedies, any kind of therapies for unknown diseases. Just recently, when there was an outbreak of the pandemic COVID-19 also, so there was people were looking for drugs, therapies from natural sources. There are so many avenues that we still need to explore, and we need to look forward to for identifying and to characterize, and then uh, help them becoming candidates for drugs. So let us talk about pharmacognosy. What is pharmacognosy? Did the slide change? Yeah. So what is pharmacognosy? Now, this is something that pharmacognosy is a science where uh, we study about the drugs from natural origin. The natural origin can be of anything, that can be plant, animals, minerals, or from microorganisms. It doesn't matter if it's only uh, plant, or if it is only animal, or if it is only mineral, all of them comes under the umbrella of pharmacognosy. This is one of the oldest branches of science that we deal with, and this essentially covers all the aspects that can lead to identification, or that can lead to uh, maybe discovery of any drug. So this term, the it was first used by Dr. John Adam Smith in 1811 in his doctoral thesis, and later there is another theory that C.A. Sadler, a medical student, he in 1815 first used this term pharmacognosy in his doctoral thesis known as Analytica Pharmacognostica. Now, essentially, pharmacognosy is derived from two Latin words. One is pharmakon, which literally means drug in Latin, and another one is gignosco, which means to acquire knowledge of. This is in Latin we're talking about. So, pharmacognosy means to acquire knowledge of drugs. Now, uh, think about think about the period of early 19th century. So there was very little exposure to synthetic drugs. I think only morphine was there. Again, morphine is something that has come from natural source, and there was some idea about quinine. Again, that has come from natural source. So there was literally negligible idea about synthetic drugs. So when we talked about we think about drugs in those era, we think about drugs from natural sources, and we can uh, we can study different historical books where our Vedas, or maybe Charak Sanhitas, Susuta Sanhitas, all of them essentially leads, or all of them essentially points out the drugs that are from natural sources. We can uh, we can actually study different drugs that have come into being from natural sources. So when we talk about drugs, the modern approach or the modern definition of drug is quite different from what we thought in the early 19th century. Uh, Currently, according to the Drugs and Cosmetics Act 1940, that actually this Drugs and Cosmetics Act regulates the complete scenario of healthcare system, or the drugs related and the cosmetic related things in within India. It was first formulated in 1940, and subsequently some amendments have been done. So according to this Drugs and Cosmetics Act, currently any medicines or any chemical substances, I would rather like to use the term chemical substances, for internal or external use for human beings or animals, and all substances that has the capacity to be used in the treatment or for the diagnosis, for mitigation, for prevention of any disease or disorder is known as a drug. Now, this drug, this whole term, it is an umbrella term. This term is an umbrella term which includes every active ingredient or any excipient that we use, or any additive that we use, any diluent that we use, that come under this term drug. Now, the current Indian scenario, there is formation of Ministry of Ayush also, so which have already included Ayurveda, Siddha, and Unani system within the modern or the conventional therapeutic systems, that is allopathy that we know. So essentially, Drugs and Cosmetics Act 1940, it includes everything that is related to drug, every chemical substances or any additive. Additives are those things which are used to make up the bulk of a drug. Suppose whenever we are suppose we have fever, so we will be asked to take paracetamol 650. Now, you have seen the tablets. It contains only 650 milligram of the paracetamol, and the other the other substances that make up the volume of the drug is are called excipients. They may be starch, they may be some other glidant, maybe mm, some hydroxy cellulose kind of things that will help in binding, or that will help in the controlling the dissolution. So these are the things, all of them come under drug. Now in pharmacognosy, the term drug is slightly modified. It is called a crude drug. It is known as a crude drug. So what are crude drugs? That is very important. Now crude drugs are the drugs that are obtained from natural sources. The natural source can be, again, it can be a plant source, it can be an animal source, or it can be a mineral source, or maybe a microorganism based source. So a drug may be known as a crude drug when they come from natural source. So they are directly collected from the nature, and they are stored without any intention of increasing their appearance, increasing their quality, or particularly increasing their purity. The thing is, whenever we are collecting a drug from natural source, we collect them. We sometimes dry them, because drying is an essential part of collection and preservation of a crude drug, because uh, presence of moisture may facilitate microbial contamination that may lead to spoilage. That is a type of adulteration that people face, that we face when we try and work with a crude drug. So one thing we may do, we after collecting the crude drug, we may dry it based on whatever type of chemical constant is present within the drug. We may opt for artificial drying, or we may opt for natural drying, like shade drying, or drying under sunlight. So we can dry them, and for storage purpose, for convenience in storing those drugs, we may cut them into smaller pieces. We may cut them into smaller pieces, and we may pack it for future use. So crude drugs are something that are directly obtained from the natural source, and they are stored after drying, and if required, cutting into smaller pieces and preserved for later use or future use. So these are the crude drugs. Now when we talk about crude drugs, this is a pictorial depiction of different sources of crude drugs. Like we can see here, that there are plant sources, animal sources, microbial sources, marine sources. We In pharmacognosy, we actually try and separate marine sources. We try and try to separate marine sources, because marine sources will contain plant, fungi, and all other in every other thing. So we try and separate marine sources, and then there is mineral sources. So essentially there are five sources which we try and we consider as natural sources: plant, animal, microbial, marine, and mineral sources. So there are some examples of drugs which have been given here. So I think all of you know about these drugs, all of you have come across the terms. We will get into this in later stages. Now suppose when we're getting a drug, there are seven methods how we can classify a crude drug. Because I have seen your syllabus, and I have seen there are classifications, there are methods of classification of crude drugs. So I will go through that line only. So there are different methods of classification. So there are seven, I think, given here, and there is another one which is very recent, and it is not very well defined. So I will talk about it in the last. So the first seven, initially I will talk about this seven methods. The first one is alphabetical classification. Now, alphabetical classification of just for this, there are seven methods of classification. They are alphabetical, morphological, taxonomic, pharmacological, chemical, chemotaxonomic, biosynthetic, and the last one I was talking about, that was serotaxonomic. So there are essentially eight methods, but I will just concentrate on these seven methods of classification. Serotaxonomy is something that is not well defined till now, so we will maybe some other time we can discuss about that. So there are seven methods. So for we will get into alphabetical classification first. So what is alphabetical classification? Alphabetical classification is just like whatever we can find in index of any books or in telephone directories, in pharmacopoeias. I think you have heard about pharmacopoeias. Uh, are you aware of with pharmacopoeia? So, can anybody just tell me if anybody has any idea about pharmacopoeias? Uh, if somebody has any idea what a pharmacopoeia is, okay, I will continue. Like just like we find how what is the easiest way to find any content within a book, that is going through the index. In index it is the topics are enlisted according to their alphabetical order. So the names starting with the words starting with A will be listed first, then the words starting with B, then this word starting with C, so so on it will go on till Z. So this alphabetical classification, it is uh, based on the alphabetical order of the Latin names or binomial names of the crude drugs. This type of classification is the oldest method of classification, and the one that we still see in different pharmacopoeias or in different books, where we have to find suppose any kind of any content from any chapter. So we will go to the index only and we will find what is the page number, in which page number it is given, we will go to that page only. So alphabetical classification is based on that only. So suppose a drug like belladonna, the uh, the scientific name is Atropa belladonna, so it will come under A. Suppose Bixa orellana, it will come under B. Suppose Cinchona calisaya, so it will come under C. D digitalis purpurea, it will come under D. So so on. Just according to their scientific name, they will be classified. It will start from A, B, C, D, and so on to till Z. The advantages of there's an advantages, it is very easy to find, but the disadvantage is you have no idea about the what it does. Suppose we are talking about the drug and we don't know what it does, it is useless until and unless we have an idea what it does to your body, how it will interact within your body, how it will react in your body. You have no use of the drug. You have to have an idea about how it will show, how it will interact, how it will give some physiological or pharmacological effect within your body. So in these classifications there is no mention of this. Again, it will not give you any idea when to collect, which part to collect, what are the chemical constituents present in it, what are the active ingredients, what are the inert ingredients, everything. Apart from the names, apart from where you can find them, it has no other use. Then uh, talking about pharmacopoeia. So what is a pharmacopoeia? Uh, a pharmacopoeia is the official publication that contains a list of medicinal drugs, their descriptions, standards, their and their instruction to prepare them and how you will use them. So a pharmacopoeia is the official book. India, the government of India, under the I think under the Ministry of uh, Ayush, currently publishes Ayurveda Pharmacopoeia of India. Similarly, Indian Pharmacopoeia is also there, published by the Ministry of Health and Health Welfare. So, Ministry of MHRD, I think. So, they will publish a book that will contain the basics, the uh, general idea about any drug, how you will use them, what are the descriptions, what are the parameters you need to see, what are the parameters you need to check, and how you will be preparing them, and after preparing them, how you will be checking their quality. So a pharmacopoeia will contain drugs according to their pharmacology, according to their alphabetical order. So the drugs starting with A will be listed first, and then the drugs starting with B, then finally it will end with Z. So in a pharmacopoeia, there are certain compositions, like you will have a monograph. A monograph, what a monograph is, uh, for any drug, suppose it is from natural source or from synthetic source, it will have the detailed description of the drug. That is known as a monograph, where you will get the botanical source or the chemical substance, chemical ingredients, the pharmacological uses, and then their identification, and then their purity standards, and then their strength. You will get those things within a monograph. Then for every drug you will get a different method of analysis. You will get a different method of analysis which will help you in uh, determining whether the drug you are using, if it is pure or not, if it is of pure if it is of desired quality or not, if it is adulterated or not. You know, adulteration is something where we mix some something that is unwanted things within your desired compound. That means you are substituting the original drug with something unwanted. They can be of anything, mostly uh, adulteration may be of two types: intentional, unintentional, based on the lack of knowledge, or whether the business the person is trying to make more profit. So based on that, you will have standards till which, below which your drug will not be accepted. Then you will get an idea about the excipients. I was talking about excipients just now. So excipients are something that are used to make up the volume, make up the weight of a particular crude drug. They are different from active ingredients. The active ingredients will only help you in giving the pharmacological effect. Whatever your body will do to the drug, that is known as pharmacokinetics. Now, whatever your body will do to the drug will be decided by the excipients only. How much longer the drug will take to dissolve, how where the drug will be absorbed, how where it should be metabolized, where it should be excreted. All of these things will be controlled by these excipients. And then again, regulations and guidelines. There are some recommendations from the ministry or from the government of India. So there, all of these things will be given within this pharmacopoeia. So for every drug, for every monograph, all of these things will be given. So there are names of few pharmacopoeia that are already very well known, like Indian pharmacopoeia, British pharmacopoeia, British herbal pharmacopoeia is there, USPNF is there, and then there is European pharmacopoeia. Then the other pharmacopoeias are there, like German pharmacopoeia, French pharmacopoeia, Japanese pharmacopoeia. All of these things are there. Then coming to Indian Pharmacopoeia, this is published by the Indian Pharmacopoeia Commission under the Ministry of Health and Family Welfare. Uh, this this particular ministry deals with appoints this Indian Pharmacopoeia Commission, and they will publish this Indian Pharmacopoeia. The first Indian Pharmacopoeia was published in 1955, and the latest edition of this pharmacopoeia is was published in 2022. Now, in 2024, there is an addendum to this pharmacopoeia. So we will, if you try and search for this Indian pharmacopoeia, so you will be getting. There are four volumes. There were four volumes in 2022 public edition, and now after the addendum, there you will find five volumes. So Indian pharmacopoeia now contains drugs from natural sources, like Ayurveda, Siddha, Unani drugs. They are also included in the Indian pharmacopoeia also. Uh, Coming to the Ayurvedic Pharmacopoeia of India. Ayurvedic Pharmacopoeia of India, it is published under the governance of Ministry of Ayush, and they appoint Pharmacopoeia Commission for Indian Medicine and Homeopathy, and they formed this. They published this Ayurvedic Pharmacopoeia of India, which was first published in 1962. Uh, Sorry, the Pharmacopoeia Commission was constituted in 1962, and further in 2006, the latest edition has been published, and during this, during every year, there are some additions to this. So right now, there are 665 monographs already there. Ayurvedic pharmacopoeia naturally contains drugs from natural sources. They also contain some about some formulations, some details about formulations like Ashava, Arishta, Kwatha, Churna, those Ayurvedic formulations, and how you will make them, what are the stability parameters, what are the standardization parameters. All of these aspects are covered in Ayurvedic pharmacopoeia. So there are, I think, 10 volumes as of 2022, and it contains around 665 of the monographs, and there are formulations. I think as of 2011, there are 202 formulations. Uh, then there is Ayurvedic Formulary of India, which was latest edition was done in 2003, and in 2022 there was an additional edition which contained veterinary uses. So you can go and search about Ayurvedic Pharmacopoeia in this link, in this web link that I've provided, and you can see that Ayurvedic Pharmacopoeia, although it is quite new to us, we are trying and include every drug, every possible drug from from Ayush sources. We call it Ayush, Ayurveda, Unani, Siddha, and homeopathy. We call them combinedly as Ayush. So we try and include all these things within our whatever we have, whatever conventional uh, system of medicine is there. We are also trying to include these things within our medicine system. Now, a monograph within Ayurvedic pharmacopoeia of India will contain these things like the title, name of the monograph, the synonyms, the description, what are the identifying characteristics like the diagnostic characteristics, then the standards like the foreign matter, how much total ash will be there, how much acid on insoluble ash, water soluble extract, alcohol soluble extract, and the assay methods, the different active and inactive constituents, again the pharmacological properties and their actions, their formulations, if there are any formulations already available in the market. So if there are formulations already available in the market, what are the testing parameters, their therapeutic indications, their contraindications, all of this, and what are the doses that the formulations should be taken in. So a typical monograph will of Ayurvedic Pharmacopoeia of India will contain all these things. Then, mm, getting into morphological classification of fruit drugs, this classification is based on the part of the plant or animal that is being used. So suppose we all know about Shorpogandha, right? We know about Raulfia. So Raulfia roots are important. Raulfia roots contain rezapine, which is essential for antihypertensive activity. We have studied this since I think class 6 or class 7. We have first came across this term rezapine and the Shorpogandha and Raulfia. Now the thing is, which part of Raulfia contains rezapine? If if you ask anyone, that person may not know it. That person may not be able to answer if it is the root or it is the leaf, it is the uh, bark or the fruit or the flower, whatever parts are there. They will not be able to particularly answer which parts we should collect. Suppose you have asked, you are in an industry and you have asked for Raulfia. Now your vendor has given you 100 kgs of Raulfia, and you see there are the roots are there, but it also contains flowers, maybe fruits, maybe bark, maybe leaves. So the amount that you wanted to receive has gone down. So that happens because there is lack of knowledge which part should be collected. So we need to collect, suppose, roots. So the roots contain rhizopin. We will only work with roots. We don't need to collect any other part from the plant. Suppose we talk about cinchona. Cinchona barks are responsible for maybe quinine, quinidine, all these things. So the antimalarial activity will come from quinine itself. Very good. So we will collect the bark only. We don't need to collect the roots. We don't need to collect the leaves. So morphological classification will help you in identifying which part we need to collect. Whenever we are talking about a crude drug, we must ensure that we are collecting the right part from the plant. We cannot be just taking a plant and uprooting a plant and taking it blindly to our industry and saying, okay, this plant, let us see which part contains this phytochemical. We must know about which part we need to collect. Now, the advantage of it is we have the idea which part needs to be collected. Again, the disadvantage, again, we have no this morphological classification does not reveal information about the pharmacological activity. uh, the chemical constituents are also not known. Maybe uh, another disadvantage, another demerit of this classification is there's a possibility of adulteration. If the drug is adulterated or replaced with a similar looking drug, suppose in case of belladonna leaves, you know about belladonna leaves, it is a very good source of atropine, L-hyoscyamine, D-hyoscyamine. Now, to maximize profit, what vendors do, they replace belladonna leaves with ailanthus leaves. Ailanthus leaves look almost similar to belladonna leaves. If somebody without proper technical knowledge and without the expertise in microscopy or maybe in chemical phytochemistry, they get this ailanthus leaves, they will think they have gotten belladonna leaves. So there is a morphological classification, there is a possibility that you may face fraudsters where they will try and replace your original drug with a superficially similar looking drug. That's a term that we use, superficially similar looking cheaper variety. So superficially similar looking cheaper variety they will give you and they will ask for the complete money. So that is a loss. Now, when we talk about morphological classification, based on their morphological characters, whether the drug has been collected from a root or maybe a leaf or maybe bark or maybe flower or maybe fruit, based on that they can be classified into organized or unorganized drugs. So organized drugs are what? Organized drugs, the term has come from organs. So if the true drug is cellular, means if the drug contains an organ, it can be called an organized drug. Suppose you are talking about Shona Pata, you have heard a very good laxative. So they will come from senna leaflets or Raulfia roots or cinchona barks or fennel fruits or isabgol seeds. So these are the organs. You can actually define them according to geometric terms. You can define a root like it is conical or it is fusiform according to the geometric terms. Whereas unorganized drugs are something that do not have particular geometric terms or particular geometric shape. So they are like exudates, resins, these come or gums, they will come into different shapes. So they will be secreted out when there is an injury. Suppose we're talking about resins. So if there is an incision on the bark of a particular, suppose, acacia arabica plant, so it will start gummosis and the gums will come out. So the gums, they do not have any particular shape. Whatever vessel you collect them in will form the particular shape of the drug. So those are known as unorganized drugs. So morphological classification will also give you an idea whether the drug is organized or unorganized. The uh, basic advantage with knowing whether the drug is organized or unorganized is you can go for processing very easily, because organized drugs, they are directly processed. They are mostly directly processed. You can just go for the extraction on them. Unorganized drugs, they need to be processed differently. maybe they will be dried first. They will be are multiple steps of extraction where you remove the unwanted things first, and then you go for the extraction. So morphological classification will give you an idea whether the drug is organized or unorganized. uh, Next, then there is taxonomic classification. Now, taxonomic classification, this is mostly done for drugs from plant sources. So you can classify, uh, you can go for taxonomical hierarchy of a plant, and you can see whether it starts from kingdom of plantae and it will end in the subspecies. So you can get into, you can take any of the terms, like you can take a family based on the kingdom, which kingdom this drug comes from. Suppose if it is a plant, it will come from plantae kingdom. If it is an animal source of drug, it will come from animalia. So based on that, you can classify that. Suppose we were thinking about drugs from this family, so you can see drugs from Apocynaceae family, or drugs from Rubiaceae family, drugs from Ephedraceae family, drugs from Gramineae family. So you can classify them based on their taxonomic position. You can go for the complete taxonomic hierarchy, and you can classify based on their taxonomical positions. You can classify based on their order, based on their phylum, based on their family, based on their genus, based on their species also. So this classification is quite flexible, and uh, there's an advancement of this type of classification, which is known as chemotaxonomic classification, which we will discuss later. So this taxonomic classification, again, it will give you an idea about the taxonomic hierarchy of the drug, where you should put it. And then the disadvantage is what you have no idea about the chemistry, the pharmacology, and the morphology of the drug. You can only know which part of the which hierarchy, which taxa it belongs to, and that's all. This is taxonomical classification. This is taxonomical classification is something that is quite old and currently almost obsolete, because we have moved on to chemotaxonomic classification, which we are coming in few minutes. The next classification is pharmacological classification. Now, this is one of the most important, although it is on the verge of maybe being not used anymore. So this is one of the most uh, commonly used classifications for during the last decade or so, last few decades or so. Pharmacological classification is something that will give you the idea which drug will give which pharmacological action. The basic criteria for being a drug is to have some kind of pharmacological action. Now, this pharmacological classification will give you an idea which drug acts on which system, or what are the pharmacological or physiological actions it will show. That's the basic premise on which pharmacological classification uh, is based on. So any drug, suppose I have there's this diagram of it. It can be of anything. Suppose a drug showing purgative action will be classified under purgative. So you will have purgatives from natural sources like senna, rhubarb, cascara. You can have cardiotonics like digitalis, quill, or stropanthus. You can have some like antispasmodics like datura, hyoscyamus, or belladonna. You can have anti-cancers like podophyllum or vinca or taxus brevifolia, or you can have carminatives like coriander, fennel, caraway, cinnamon, anything. So if tomorrow a new disease is discovered, and for that disease a drug from natural source is also discovered, so that will be under that new class of drugs. So it is unending. This is always evolving, but somehow it has some major flaws that I'm going to discuss now. So in pharmacological classification, the basic advantage is you have the idea uh, where it should be used. Uh, in ancient times, when we did not have all the means to identify drugs like morphology or the suppose alphabetical classification, we only relied on pharmacological classification. Uh, I think you have heard about the discovery of Cinchona. Uh, I will just tell it once more. Cinchona, this is native to Peru, the South American country of Peru. Right. So during that time, during early 17th century, Peru was colonized by the Spanish people. So there was in this region there was a count of Sincon who was ruling this region. Now his wife got fever, and the whatever the healers were there or the doctors were there, they were unable to treat them. So the the people around that particular region, they started say this once they got to know about the fever, they suggested the count to take his wife and make her bath in a pond nearby. So that was a lake, not pond, a lake nearby. So that lady, that countess, went and took a bath in that for a few days, for 7 to 8 days. Continuously, she took bath in that lake, and miraculously, her fever got away. So the people were more fascinated with the fact that just taking a bath in this lake will cure fever. So they started snooping around the lake, looking for whatever the reason behind that. So they found out the pond was surrounded by cinchona trees, and the cinchona trees, the barks, the leaves, all of them, when they dry off and they will fall into the lake. So the lake, the water of the lake was not contaminated. Let's use the term. Uh, it was like a solvent to those to the anti-malarial ingredients from the lakes. So the Jesuits were the Jesuits were the healers from Vatican. They went there and they collected some sample of this cinchona bark, and they came back to Vatican and they stored it for future use. Now, after 16th century, everybody forgot about it. In During World War One, there was an outbreak of malaria. This During this time, the the people went to Vatican for support. They asked for healers, they asked for nurses, and then Vatican actually revealed the existence of cinchona bark to the community, and only then cinchona came into existence. So the initial identification or the initial discovery of drugs were based on this pharmacological classification only. It was not based on any other scientific reason. It was only based on the pharmacological actions a drug may exert on a body. So that is the advantage. Then what are the disadvantage? There is in our pharmacy we study something called structure activity relationship. We uh, we study that the structures of any chemical are responsible for showing activity. This is known as structure activity relationship. Now we know about the activity, but we don't know about the structure. This is a major flaw of pharmacological classification. Then it, you can see cinchona bark. You can see cinchona bark, which contains quinine and quinidine. These are enantiomers of each other. Whereas the basic difference is just see that there are two chiral carbons at 8 position and 9 position. So the so in case of quinine, it contains 8s. The configuration is 8s and 9r. Whereas in case of quinidine, it is 8r and 9s. Now quinine is a strong anti-malarial drug, and quinidine is responsible for showing anti-arrhythmic activity. Arrhythmic activity is inconsistencies in the heart rhythm of heart. So these two chemicals come from the same drug, that is cinchona. However, quinine and quinidine, they show polar opposite activities. They show complete different activities. So where will you put cinchona as a drug, under anti-malarial or under anti-arrhythmic? This is the biggest flaw pharmacological classification has. So we have moved on from pharmacological classification, and the most widely accepted classification now is chemical classification. Now, this chemical classification is based on the different types of chemicals that are present within a crude drug. Now, just now, I was talking about the different ACR thing. So ACR is the structure of a chemical compound will decide how the chemical will act or interact with the body. That was the basic idea behind ACR. Now, every chemical are so diverse, they are structurally so diverse. Uh, we can classify them according to their structural features. The thing is, these chemical substances or active constituents, as we call it, they are nothing but metabolites of plants. Suppose a plant has consume some food, suppose nutrient, and it will break it down into starch like compounds, and then the starch is again it will undergo different glycolysis or Krebs cycle, and it will form pyruvic acid or acetyl CoA as end product. And this acetyl CoA or pyruvic acid will undergo different uh, different reactions to form uh, metabolites. These are known as secondary metabolites. We in we try to classify drugs based on their chemical classification, specifically on the type of secondary metabolite that is present. The drug that we are using, it will contain some kind of secondary metabolite, or in some cases primary metabolite. So based on the chemical nature of them, we will be classifying them. So the advantages, it is most widely used because the chemical nature will give you idea about a lot of things, because the only thing we are looking for is its pharmacological activity. So if you have the structure of a drug, you can assume, you can predict, you can actually deduce the pharmacological activity it will show within a body. Now the same goes for, suppose a drug that is showing some toxic effect. We can actually modify the structure. We can go for semi-synthetic modification. We can go for semi-synthetic reactions, and we can modify the structure, and we will be able to uh, remove the portions that are showing toxic effect. We can include the portions that are beneficial to the activity. So that is an advantage that we get with chemical classification. Now, chemical classification, there are again some disadvantages, like suppose there are drugs, because of their complex metabolism, they may contain more than one type of chemical constituent. So if a drug contains more than one type of chemical constituent, like if it contains an alkaloid, or if it contains a glycoside, or if it contains a tannin, it becomes very difficult for us to classify them with under a particular class. An alkaloid, cinchona cannot be classified as a alkaloid containing drug, because it also contains glycoside, it also contains some amount of tannins. For that, to overcome this, we have come with a different solution. We have taken uh, active constituents and inert principles. Active principles or inert principles. So, what are active principles? Those principles or those ingredients which are biologically active, like they will be showing some biological effect on the body. So, these are the active principles. We have some examples here. So, based on the type of active principle present within a drug, we can classify them in chemical classification. So, suppose chemical the active principle in cinchona is quinine, so we can call it as an alkaloid containing drug. Suppose the digitoxin is a steroidal glycoside, so digitoxygenin is the active constituent, so we can put digitalis under glycoside containing drug. Similarly, terpenoids like menthol or maybe umbelliferones, sorry, anithols, fenjones. These are like uh, terpenoids or essential oils. So we can put them under terpenoid containing drug or volatile oil containing drug. These are the active principles which contain some kind of, which will show some kind of biological activity. The next one is inert principle. So what are inert principle? These are the chemical substances which will not show direct biological activity. These will not show direct pharmacological action, but it will play a special role in the stability, solubility, or preservation of the active constituent. Mostly, we can call them as excipients, additives, or as diluents, like starch, lignin, fixed oils, gums, mucilages. This gums or mucilages does what? They will help in providing with the binding facility or the emulsifying property or as a stabilizer. So if you are preparing a herbal tablet, you can use a gum like gum acacia or trakan, and it will, those gums will act as binders. Suppose you are using starch within your tablet. Now starch doesn't have any pharmacological effect, but starch will help you in making the bulk of the drug or the volume or the weight of the drug. Then there is cellulose or hemicellulose. You all know about cotton, tulo. Cotton doesn't have any pharmacological effect, but any therapy, any surgical therapy is incomplete, or the cotton use of cotton is very important in surgery because it helps in wound cleaning, suppose dressing of wounds. So cotton, although it does not have any pharmacological effect or it cannot be considered as an active principle, it can be uh, it can be very important in therapy. For these things, these inert principles, we call them as pharmaceutical aids. Pharmaceutical aids which do not have pharmacological effect, but they will somehow help in therapies, or they will help in formulation of uh, different uh, different form. They will help in different formulations. Then I was talking about what was the problem with chemical taxonomic classification. So we came up with a different type of classification that is known as chemotaxonomic classification. Now, chemotaxonomy is something that it is a word that has been derived from two words, chemical and taxonomy. Now, this chemical and taxonomy, this the basic idea behind this classification is a particular family of plants which will contain similar type of chemical constituents. This is the basic idea behind this kind of classification. Uh, we can be very sure about some family like Solanaceae family. Solanaceae family, like Atropa belladonna or Hyoscyamus niger. Now see, all of these plants in Solanaceae family, they will contain tropane alkaloid. Tropane alkaloids are what? Tropane alkaloids are these are bicyclic compounds. They will contain a pyridine and an imidazole ring. So they will contain sorry, tropane alkaloids will have a pyrrole and pyridine ring. So they will contain what? They will contain the tropane alkaloids, Solanaceae family. Similar for that, uh, just look at this, like isoquinoline alkaloid. Isoquinoline ring, isoquinoline alkaloids will be found in Papaveraceae family. So any plant of Papaveraceae family will contain some amount of isoquinoline alkaloid. Similarly, for Rubiaceae family, they will contain some amount of quinoline alkaloid like cinchona, and they will have maybe more, maybe less, but they will certainly contain some amount of quinoline alkaloid. Similar to the those like terpenoids for Asteraceae family or Fabaceae family, they will have carotenoids. Rubiaceae family also contains some amount of iridoids. Now, the basic advantage of this is, suppose you have got a crude drug. Now you have no idea what drug it contains. It has come into in a powder form. So your job is to find out which plant it belongs to. So first you will go for microscopy. If the microscopic evidences are enough, good for you. But if the microscopic evidences are not enough, so you will go for chemical identification. The chemical identification will give you an idea about what type of chemicals are there. You will go for extraction, then you can isolate the drugs, and you can go for spectroscopic analysis or chromatographic analysis. You will be able to elucidate the structure of the compounds. Once you get the structure of the compounds, you can reconnect the dots that will lead you to the basic family of the plant, and from that you can actually identify the, you have done the quantitative analysis also, so you can identify the genus and the species also. So you will be having a set of documentary evidence on the uh, microscopic characters, the physical characters, as well as on the chemical characters. So they are enough to for authenticating any plant. That will help you in identifying. Okay, so I have been told this is a Atropa belladonna. Let me see if it contains some amount of tropane alkaloids or not. You can see you will find, okay, this contains a very small amount of atropine. So you will try and understand whether the amount is related to Atropa belladonna or Hyoscyamus niger or any other plant of Solanaceae family. This is also very important in identifying the different markers of a compound. Suppose in herbal drug industry, when we talk about standardization of herbal products, we always go for markers. We always go for biological markers, and these compounds, these chemotaxonomic markers, these chemical compounds always act as biological markers which help us in identifying whether our drug is pure or has been adulterated or not. And then the last one that we'll discuss is biosynthetic classification. Again, biosynthetic classification is something that I was just talking about in the slide on chemical classification. These are nothing but different metabolites. Now, this metabolism takes place in different pathways. They will come from different pathways. Different biological reactions will take place. Biotransformations will take place, and from suppose from acetyl CoA, you will get steroids. Uh, uh, These are the biosynthetic pathways. There are certain biosynthetic pathways for terpenoids also, like acetate malonate pathway or deoxyxylulose phosphate pathway or methyl HCl pathway. Then there is a shikimic acid pathway, which is responsible for biosynthesis of different aromatic amino acids. Then there is anthocquinone glycosides, like there will be Sena, the chemical constituent of Sena is known as senoside, which is an anthocyanin or anthocquinone glycoside. This anthocquinone glycoside will come from malonyl CoA pathway or acetate malonate pathway. So these are the different pathways that will give rise to the different chemical constituents, and based on the different pathways or different starting materials that we encounter, we can classify them. Just like this, like propane alkaloids, they come from ornithine. Or suppose a drug like conine will come from lysine. Or suppose morphine or codeine, the alkaloids from uh, opium, they will come from tyrosine. They come from tyrosine alkaloid. So these, they will come through different and very specific pathways, biosynthetic pathways, and based on that we can classify them. The advantage of that is we can identify the path. If we can identify pathways, we can uh, alter the rates of reaction, and from that we can actually alter the chemical compositions of that will go into um, plant biotechnology, and from that we can actually manipulate the different steps within a pathway. From that we can actually uh, increase or decrease the chemical constituents. Then the last one I was talking about, that was serotaxonomic pathway. I have not put it here, so I will just have two lines for serotaxonomic pathway. So serotaxonomic classification of products, it is done on the there are certain proteins that can be found from plants. So these proteins are collected from the plants and they are injected into the test animals. Now these proteins will act as antigens, and in response to those antigens the animals will produce antibodies. Now, based on these antibodies, which kind of antibodies are being produced upon introduction of those antigens, based on those there is a classification that is known as serotaxonomic classification. Like, if uh, two animals produce similar antibodies on being injected with two different proteins or antigens, you can say, okay, the or the proteins come from similar or very closely related compound, very closely related plant species, or very closely related taxa. So from that you can classify them. So we go for serological studies there, and we can the serological studies based on the plants are known as serological studies, which is quite new, and I think in future it will be more explored, and the findings will come forward.
Yeah, 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 audible. So the session is open for cross question by the students. The discussion session. So I will request our student to ask some questions if they have any. তোমরা যা কিছু কোয়েশ্চেন জিজ্ঞেস করতে পারো, প্রবলেম নেই, স্যার অবশ্যই তোমাদের অ্যান্সার দেবেন। লজ্জা পাওয়া বা ভয় পাওয়ার কিছু নেই।
ফোর্থ সেমিস্টারের স্টুডেন্ট যাদের আছে
হ্যাঁ,
তোমাদের
হ্যাঁ,
ফার্মা,
হ্যাঁ, ফোর্থ
তাদের
সেমে তোমার
যদি কিছু
সিলেবাসে আছে।
ডাউট থাকে, ডাউট
হ্যাঁ,
থাকে,
কোনো ডাউট থাকলে কোয়েশ্চেন করতে পারো তোমরা।
আমাদের এবার ফার্মাকোগনোসিস সিলেবাসেই আছে। তার জন্যই স্পেশালি এই প্রোগ্রামটা রাখা হয়েছে তোমাদের জন্য।
তো যদি
অভয়
কোনো
বাবু
কোয়েশ্চেন
আছে,
থাকে তুমি।
তাহির আছে।
দেবজিৎ আছে এখানে, দেবজিৎ, সম্রাট আছে। Because actually the topic was it's a very difficult topic to wrap up within 1 hour.
Yes, yes, yes.
So exactly. So it was a 1 hour session with 31 slide, but information of almost 310 pages. So, we were our students were very fortunate to witness this lecture and to listen to this lecture. I think they are very much in it, and so much in it that they don't have any kind of question.
No, actually, we teach this in our fourth semester also. So the things that I have just included in this slide is for 3 or 4 classes, and those are for 4 or 5 hours at least. So it may be a bit overwhelming sometimes. Hello, hello.
আচ্ছা, দেবজিৎ has raised his hand. Devjit, you can ask if you have any question.
Sir, will you please describe about the Siddha and Unani drugs?
Okay, Siddha and Unani drugs. These are different systems of medicine. Suppose we know about Ayurveda, right? Ayurveda is ancient Indian system, although it has origins from Middle East also, because it has come from maybe even before the Aryans. So Unani and Siddha. Unani is a system that was originated in ancient Greece. There were two. I think two for two paths, like the Pythagorean one, and then there was, I think, the Plato, or someone like that. So they had their own system of medicine, and this there was this Greek system of medicine was going on, and then there was Persian invasion in Greece, right? There was Persian invasion in Greece, and they somehow they somehow collected some part of those Greek uh, scripts from the during their invasion, and when they came back to Persia, there was this amalgamation of their own Persian system of medicine and this ancient Greek system, and from that this new Unani system of medicine has evolved. So this is no Unani system of medicine. It's a blend between Greek system of medicine and this Persian system of medicine. And as for Siddha is concerned, Siddha is purely Indian system of medicine that was originated in the southern part of India. In Bengali, we call something called Siddha Purush, right? So Siddha Purush is কাকে বলে? Siddha Purush যে মোক্ষ লাভ করেছে। তো in Siddha, there was this practice of dhyan and this moksha love, and whoever got moksha was known as a siddha purush or siddha. So তাদের যে their system of medicine that is known as siddha. So these are completely these are two very different system of medicines, similar, and in India, currently we have Ayurveda, Unani, Siddha, and then there is homeopathy. And if you go out of India, you will see there is traditional Chinese system of medicine, which is very popular. So what we are trying to do is we are trying to get these things back. We are trying to revive this system of medicines. So they will be beneficial. So Siddha was basically based on the meditation, yoga, and stuff like that. So uh, it mm, focused on uh, natural, I think natural energies like rain or maybe storm, these things. So they started praying whenever someone got sick. They started praying those to these energies. So that is Siddha for you. And anyone else?
Okay.
I think you have got your answer, right?
Anyone else have any other question?
Anyone
কেউ
else?
করতেই পারো।
তোমাদের এখন কোয়েশ্চেন থাকলে তোমরা করতে পারো। Okay. তো সন্দীপ, ক্যান উই, আশা করছি কারো কোনো কোয়েশ্চেন নেই তো।
Yeah, we can. Yeah, you can stop the session. Thank you so much,
ওকে
sir.
ওকে ওকে, থ্যাংক ইউ
Actually,
সো
I'm
মাচ স্যার।
traveling right now,
Thank
And
you,
I'm
sir.
so you'll
also
get a
outside right
lot
now,
of
so
background noise.
thank you so much, sir, for your precious time.
Thank you, sir.
And
It was an honor for me,
this
sir.
this
Student,
lecture
please
was
don't leave. We have some topic to
exactly.
discuss with you. Student, please don't leave. Huh?
Yeah, I've given. I think there was my mail ID was given in the first slide. So if anybody
Yes.
can wants to uh, connect, please feel free to connect. There's no nothing I I wouldn't share with regarding pharmacognosy. So,
Sure,
thank you,
sir.
sir.
Definitely, definitely, sir. Thank you, sir. Thank you so much.
Thank you.
Thank you. Thank you.